Hello, I'm Neil Spooner, the chair of the Patient-Centric Sampling Interest Group, and I'd like to welcome you all to our workshop today. It's a new era in clinical diagnostics, the power of self-sampling, and this is part two. So we had part one a week ago, focusing on the challenges to self-sampling, the more widespread adoption of self-sampling for North America. This week, we very much like to get focused on the solutions and next steps. So we shouldn't be any longer talking about the challenges. We want to talk about what the solutions are to bring these technologies forward to more mainstream use for clinical diagnostics, and then how we go about that. So just a little bit of background for everyone. Patient-Centric Sampling Interest Group is supported by sponsors. And so we'd like to say thank you to all of those sponsors that enable us to run this workshop today, run our website, run all of our webinars and all of our activities and working groups, without whom we wouldn't be able to do all the fantastic things we do together. Today, as I say, the challenges were set out in the workshop on the 30th of April and have already been shared with the PCSIG LinkedIn group for comment. So the aims of today's workshop is to start identifying solutions to the challenges presented in part one of the workshop and set out a plan for delivering these solutions. Now we realize we've only got a 1 hour workshop today and there's a lot to talk about. We know we're not going to get everything done. So the idea is this is just step one. There will be ongoing activities which we'll talk about towards the end of the event, which we're going to invite all of you and others that aren't here today to take part in. And we'll also be looking for other organizations to partner with who are best placed to help us move forward in this. But before we go any further, I'd just like to introduce today's, just a minute. I'd like to introduce today's panelists. So I'll just minimize the slides for a moment. So if we can go in order, Brad, can you introduce yourself first, please?
Sure. Brad Collier. I am a director of research and development at LabCorp. Been working with microsampling capillary blood collection devices for coming up on a decade.
Thank you, Brad. Marianela?
Hi, my name is Marianela Perez-Torres. I am the director for the Division of Chemistry and Toxicology Devices in the Office of In Vitro Diagnostics. At FDA, our division regulates the blood collection devices that are used for clinical testing, and other sample devices are also regulated by our office.
Thank you, Marianela. Sharon, could you introduce yourself, please?
Yes, thanks for coming. I'm a pediatric pathologist and a hematopathologist, and formerly directed the pediatric laboratories at the Children's Hospital at Stanford, and then co-directed the adult pediatric services after that at Stanford University. And I'm currently the IFCC chair of microsampling group as part of the Emerging Technologies division of the IFCC. And I also serve as an advisor on the FDA's medical device panel.
Thank you, Sharon. And last but not least, Bryan, could you introduce yourself, please?
Certainly. Bryan Andrus, CEO and founder of US Specialty Labs. Been working with microsamples and patient-collected samples since 2016.
Thank you, Bryan. And just from part one, we had a patient representative, John Fabule, but he's unable to be with us today. But hopefully he'll be involved in the ongoing activities after this event, because obviously we're talking about patient-centric sampling, and the patient voice is very, very important here. So just go back to sharing the slides. So with this panel from discussion, we've got some topics that we agreed and that were shared with the LinkedIn community over the last week. But we'd like you, the audience, if you've got any suggestions, comments, please use the chat function. As I said, we're about about solutions and next steps here. We're not about challenges. So we're now moving into the positive space of how we move forwards together. Note, this workshop is being recorded and will be available on demand after the event. So let's start on the challenges. So I want to start really with the patient. So patient and provider need to be confident in using the tests and the results. So how do you raise that confidence that the patient can use these technologies and therefore we can generate good quality data that can be used by clinicians and others for decision making? What are the possible solutions to this concern about confidence in using the technology? Has anyone got any suggestions on this at all as to how we move forward?
Yeah, when I think about this, I start first with, thinking about the collection of the sample in the field. The analytics, as laboratorians, we all have a good handle on and what needs to be done. It's appropriate. But you put some of these technologies in lay people's hands, and sometimes the quality kind of falls apart a little bit, and that's a known problem. And so you need to make sure you give them the appropriate resources and confidence. Everything from videos to hotlines perhaps about how to do things may be necessary, but making sure that they generate a high quality sample at the very beginning to set up the whole process for success.
So would one of the solutions be to have some sort of standard of what's expected in those materials and training?
I mean, it would be nice that, there's a lot of different technologies out there, especially when you think about capillary blood. There would be some sort of standardization in the process so people have a general idea of what to expect when they're collecting capillary blood, for instance. Now, when you move from matrix to matrix, that's a little bit difficult. But when you're providing the IFUs, you can make them too burdensome for the patient. People will just, look at it, see there's too many steps. And so there's a balance there. And if you can, standardize it to some degree across the different technologies, that really would help, I think, on the uptake and use.
I think the use of different media as well. Several of our channel partners are starting to expand the role of the online interaction when people go in to register kits, and are including different types of videos, are including imagery of good and bad samples to prevent samples that may come in from being rejected, reinforcing the kind of nuance via the video and in text, the way that samples need to be collected in order to have it bring in. It stops and starts with the quality of the sample, and that we can't compromise on that. I think also making sure that there is care taken to the way that the samples are being returned, so that there are not challenges, or the stability is not impacted if they are not being returned in a timely or appropriate manner, meaning quick enough or cool enough in some cases.
Yeah. Any other comments on solutions on this?
Neil, I just have two comments, and the first comment is, if I were to ask the audience one thing, I would highly recommend going back to the webinar we did a week ago to see John Fabule's talk. I have to say, I've never really heard a talk like his. He speaks to all the issues beautifully as a patient. And it's really a very amazing talk, not to be missed. After you see it, it's very unforgettable, in my opinion, and just highlights the engagement of the patient in what Brad and Bryan were talking about, the patient's voice. And I know the Agency for Healthcare Research and Quality has advocated for patient education materials to be written between the 4th and 6th grade of reading literacy. Many of us talk about, healthcare literacy, but there's a real need to attend to actual literacy. And that's what their recommendations are for the patient educational pamphlets. So I would just say that manufacturers can also follow suit, because many of the pamphlets are actually really quite sophisticated, and their target audience may not be able to handle that level of sophistication in the pamphlets in English.
Yeah, I think that's a really good point, Sharon. And to me, one of the recommendations for going forward, as well as developing a standard, is to make sure that when you're developing these standards, that we involve patient groups in doing that. All too often we forget that, and we build something that we all think works, but as you say, it's too complex for those that are actually wanting to use it. And if we fail at this first step of collecting a good quality sample, then all the sophistication further on down the line is worth nothing.
Exactly.
Okay.
So, yeah,
Oh,
I mean, obviously, we all talked about the patient's comfort with doing all of this, but there is a second piece to this here, the provider as well. So that, I think, comes back to more of the scientific rigor of investigating, kind of what Bryan was leading to, making sure that not only you collect an appropriate sample of appropriate quality, but it gets into the lab in an appropriate manner, and that the analytes or the matrix you're using are the appropriate matrix. And so it comes back to perhaps being a little transparent to at least to your providers that you're offering these to, and saying, we have all the quality controls in place. We believe this is a high quality test, and here's the data to prove it.
Yeah, I like that. Any other comments around this bit? And I think a lot of these things are going to be intertwined as we go through. It's not simple, but this is a really good discussion. Any other comments on that patient piece?
I would say, I think it was Bryan who mentioned before, not all devices are created equal. So making a standardized collection device procedure might not be the type of standardization you're looking for, but rather the characteristics of the instructions that you're looking for, the simplicity part, the mechanisms that manufacturer will put in place to ensure that anybody that receives and decides to use these devices can do it successfully.
Yeah. Yes, agreed. Okay. Anything else on that patient piece before we move on to other aspects? Just a comment from Nirav: cannot agree more. Burden of patient has not always been met, especially having watched at-home testing in my own home for 25 years. So thank you, Nirav, for that supporting comment. The next thing we've got, regulations. We're going to just jump over that for now. We're going to come back to that towards the end of the discussion, because we want to get into the meat of the challenges around the lab and how we come up with solutions there. So, as we've seen here, some of the challenges were validating a system, not just an assay, more complex than just developing an LDT, off-label use, analytically what's good enough. So these are some of the challenges we talked about last time. People, it'd be good to start hearing some of the solutions here. Has anyone got any suggestions for how we can move forward with solutions?
Well, I mean, I can speak to from our standpoint, and this touches a little bit on the patient/provider as well. We do have, when we have new people that we're bringing on board, we're very transparent with respect to the validation process that we go through, the thermal cycling that the samples are subjected to, the vibration the samples are subjected to, and we specify all of that. I think it was actually Marianela that brought up the fact that this actually is a system, which is very true. And the system is only as strong as the weakest link. So if it is poor transport, that's as bad as if you get an incomplete collection, because the sample will not be be rendered useless or not usable. And I think that as those of us that are providing these types of services, we need to be very transparent, very open, and to the extent publish some of this. I don't think the market is saturated enough to where we should be concerned about sharing some of the best practice. I think it's okay for us to be upfront and say these are the guidelines, or these are the processes that we are following. If somebody's got suggestions as to other things we may be doing, we welcome it. We've had some of our clients send in very, very smart people who have given us suggestions, and we've incorporated those.
That's a great suggestion. Is going back to the solution for the first one, is there further standardization of the kinds of tests and acceptance criteria needed, or is there enough standardization out there? We just need to publicize it more.
There are, acceptance criteria out there it's analyte dependent. There are some acceptance criteria out there, but there's not always agreement about which ones. There's a lot of information out there. That may be difficult to come to consensus on what's appropriate, because there's nuance to a lot of these different things. And on top of that, people have different opinions. I think, yeah. I just think it would be difficult to come to a consensus on analyte-dependent markers. That being said, there is a question of, what is good enough? And that is a very difficult question to answer. I'm actually interested to hear what my colleagues have to say about that.
If I may, I think the challenges here is that you are having in this list challenges for the lab, that if manufacturers of the devices, of the collection devices and the assays come together, it's less burdensome to the labs, because the labs just have to verify what the manufacturer established. So, and they can focus on the more clinical lab challenges instead of trying to invent the wheel. So it's related to the challenge to the regulatory part of it. If the devices, the collection, sample collection devices are medical devices, they need FDA authorization. If we start there, authorizations that include use very broadly for self-collection, such that labs can implement such devices with tests that are also FDA authorized for such uses, that will lessen the burden to the labs regarding their verification activities.
I agree. I think that's, as you and Bryan said earlier, it is a system, and the more clearances that components of those systems have, it makes the approval for the lab to verify and validate that system much easier.
Yes.
Okay, how do we go about that then? How do we get to that place?
The manufacturers.
I think the run up to this, the conversation that was had regarding someone convening a forum where folks would be able to have those types of discussions and try and figure out how to go about doing that to chart a path forward. Marianela, as you indicate, FDA cannot lead that, but you can participate, which would be very, very welcome. And I think there is, certainly on the panel here, I think there is very strong interest for having that type of a meeting come to place, come together.
So we're talking about the collaborative community, yeah?
Yeah, I mean, I think you use the Crystal City meeting as a, as kind of an analog.
Yeah, and that was something in my background from drug development in the pharmaceutical industry, validating bioanalytical methods. As I described to you earlier, 25 years ago was a bit of the Wild West. So industry and regulators came together at Crystal City Hotel in Maryland to talk about this. And over the subsequent 20 years, there were more meetings, more publications that ended up with an international standard that is now followed by every regulatory authority around the globe. And so labs kind of know what standards to work to wherever they are in the globe. But hopefully we can do this in a quicker time frame than 20 years. But yeah, okay, that sounds like a great solution. Sharon, were you about to say something?
Well, the idea of a collaborative community, launching this, what seems like a new era truly in diagnostic testing, although we do have, the model of diabetic self-testing, and there's more out there when you really think about it that we have that's foundational. But in the meantime, I know that LabMed UK is coming out. They've written a proposed sort of guidance document about this capillary versus venous and so forth. That is, I believe it's under review, should be published.
It was accepted today, Sharon, so it should be.
Oh my gosh, okay. Well, thank you for that, Neil. If anyone should know, it's you. So Neil is the most connected person I think I've ever met. But anyway, Christophe Stove in Belgium at the University of Ghent has written more than anyone, I think, in the world about this in this arena, tremendous depth, and he's been involved in some guidance documents. And so it's really good to see these various groups and teams putting things out there. There's a lot of publications on some of the successfully authorized, FDA authorized systems, if you will. And so there's really a lot to look at and to learn from. There's a lot of webinars on Neil Spooner's, who's, I think, he founded this PCSIG, and there's a lot of webinars. There's a lot of educational resources already, but I think the collaborative community would sort of bring us to the next level and allow for a lot of information sharing.
So I have a question about the collaborative community. Would it be too difficult for the collaborative community to involve voices from outside the United States? So regulators from beyond the United States, companies from beyond the United States, or do we just need to have to separate these conversations into different parts of the world?
Everybody's welcome in the collaborative communities as long as the leaders of the community establish what the membership is. But basically, it's a forum where everybody from the private and public sector could convene to work together on medical devices challenges. They can be from other agencies, from other places. Obviously, because it's related to the FDA, the focus is the use in FDA regulated products. However, we do collaborate with our international colleagues. So and we continue to look forward for common grounds to make the regulatory part easier for global companies to adopt. So it's not, I don't think it's limited to U.S. Populations when they come together.
Because the example of the Crystal City I was given in the pharmaceutical industry, Marianela, was, it was hosted close to the FDA, because we all know the FDA, it's difficult for them to travel. So it needs to be in a hotel somewhere near you guys. But actually it covered regulators globally. And as I said, it turned out to be the publications that came out of it and the ICH guidance is international. And it would be great. But what I'm asking is, do we think it would just be too hard with different, Europe going one way, MHRA going another way, FDA going another way? Would it be too hard to get everyone in a room, or is it worth trying?
Yeah. Oh. To the group side.
Okay, we should try. Okay, I like it. We have to be ambitious.
And again, also for these communities, it's not that they have necessarily deliverables to provide to the FDA, right? It provides, it can change over time, and the goals can be modified accordingly. So the first step is to get together with everybody to actually, and you have already doing some work before that assignment, because you already have some the issues identified, the solutions you want to find. So that gives you already a framework to facilitate the next steps.
Okay. I just want to say to the people, and Nirav, I apologize, I can see you posting a lot. Just let everyone know that we are capturing all of the comments. We won't necessarily get to read them all out during the hour that we have together here, but we are capturing them all. All the comments will be summarized in the summary documents that will come out of this event and potentially in the publications, and will be brought forward into future discussions, etc. So please, if you have a point, please don't get frustrated that we're not reading it out. We can only deal with so much in an hour. So please, if you've got other things to say, please just type it in the chat, and we can take account of it after the event. The panelists from Europe and North America will be meeting in the next few weeks to talk about next steps. And then we will be inviting you all to come back together to participate in agreeing on what those next steps will be, and then participating in those next steps. So this is intended to be an inclusive, forward-looking thing. Just apologize if you feel like you're being ignored a little bit, Nirav and Gloria. So is there anything else we can say on this particular thing about the labs? Are there other solutions we can home in on other than getting us all together in one room? And the manufacturers standardizing and having a lot of the things ready to go for the labs was another good suggestion. Anything else we're missing there?
Well, to go back to the what is good enough, I think labs that offer this kind of testing need to be very clear on what their intentions are for the testing. A lot of times these types of things are labeled as not for diagnostic use, but it's not very clear to the patient. And so perhaps it's screening of some sort, but it just needs to be abundantly clear what type of test is being offered and what the content is.
And that's really good, bringing the patient back to the center there as well. There's no point doing this if it's not clear to the patient what the value is. That's a really good point. Let's just scroll down to the next piece. So logistics and data management, is there anything separate there to what we've been talking about? Some of this is outside the lab, how we get samples, how we get the devices to people's homes, how we get samples to the labs, how we get the data to the right people. So it's kind of related. Could it be solved in the same forum that we were talking about, or are there additional other solutions here as well?
I think the question about data, that's going to be specific to the use case and to the ecosystem that is involved. I'm not sure, that you could, fully define something on that. I think in terms of the, getting the sample or getting the kits out to the people, 3PLs are very skilled at being able to do that. I think, bringing the kits back home, back to the lab again goes to what I touched on, and what Brad and I feel strongly about, I know, is making sure that the transportation of the sample is done in such a manner that it renders it useful, and that there are not divergent acceptance or rejection criterion on the part of the lab, which sadly does exist in some cases today. That people are following good laboratory, good scientific principles with respect to the way that they specify samples need to be returned to the laboratory.
And I think also when we think about the system, going back to that kind of thought process, you may also want to consider your packaging solution. How are you getting samples back into the lab as part of the process you're validating? I know, Brian, you've mentioned about making sure that things get back at the right temperature, but also you've also mentioned like dropping things and other ways that the package can be perhaps abused a little bit. Everyone's received a box that's had holes in it and wondered what happened there, kind of thing.
Yep.
This is also an area where I think we can go global in terms of looking at best practices. I know that in our European webinar, which is also on the website now, that preceded this one, Vincent Molnar talked about the Netherlands having a separate postal service for this very purpose, which I found very interesting, and I'd like to know more about. The same way, there's an NHS program in the UK that is super interesting, which has validated urines that are both home collected as well as posted to the lab through the postal service. And they validated that for exploring C-peptide levels throughout Scotland first, to look for endogenous insulin production, and to basically find patients that have been misdiagnosed, that have young onset diabetes, but that are actually generating enough insulin that they can take tablets rather than face a lifetime of injecting insulin. And so there are these best practices that exist, and one of the advantages of going global, I think, is that we can compare and contrast and learn from each other.
Yeah, and I think encouraging people to publish their data as well is really important. How many labs have got really good data that they're just not sharing that would help all of us?
Right.
And there's a fabulous new journal that people will hopefully be interested in publishing their data in, the Journal of Patient Centricity, which is brought out in association with the Patient-Centric Sampling Interest Group. It's a fully peer-reviewed open access journal launched just in the last few months. First few publications are in there. People were always asking us where to publish their patient-centric research. Well, now there's a fully peer-reviewed home going through a reputable publishing company. So hopefully we'll be able to get a lot more publications of data, which will help us all. Anything else on logistics and data management at all?
I think kind of going back to manufacturers and thinking about logistics in the lab, these are small volume samples, which can be a bit more tedious for the lab to deal with at times. So I think working with manufacturers, as Marianela kind of alluded to, with how to handle, automate these samples in the lab would be a great piece. Making sure you don't get, sample spillage or sample wastage. It is a lot smaller samples, which is a good thing, but it can be a bit frustrating working with them at times. And so not just the logistics of getting samples into the lab should be considered, but the logistics of handling the samples in the lab.
Okay.
Yeah, the laboratory workflows are definitely much more challenging than normal samples coming in. You're essentially one and done. You've got to get it right. And it can be challenging. It's a very manual process typically.
Okay, cool. No, I'm liking these kind of solutions. I mean, a lot of them involve people sharing and talking to each other is what we're coming to the opinion of, and it's just figuring out ways of how to do that and then how to share the output from those conversations. And I think that's the important thing. I think there's been lots of conversations going on in different corners of each country separately, and bringing a lot of this into one room to learn from each other and share best practice, as Sharon was talking about earlier, is absolutely a perfect way forward. And I don't know whether you think, Sharon, that IFCC would be interested in helping to take a lead in some of just helping to bring the different parties together.
Well, I can't speak for all the IFCC, but I do know that the chair of the Emerging Technologies Division, Damian Grusin, is all about collaboration. And so, given that, I think, it sounds like a really positive proposal. And I think there's a lot of people probably, or at least some people on this webinar that work in pediatric environments, and they are probably all very well acquainted with the increased labor time it takes to handle small specimens, the aliquoting, the amount of time doing that can lead to occupational issues like carpal tunnel, and it's not trivial. And so I think that often brings you to the cost benefit, which is a thorny subject.
Yeah, well, that's kind of where we're going next. Before we do go there, we've got 20 minutes left. Any other comments on logistics and data management at all? Okay, so let's jump into economics and, cost benefit models. Obviously, some things are very country dependent. Reimbursement, North America, USA has a very different model compared to a lot of other nations around the world. But a lot of other things are in common. Everyone, regardless of who's paying, has to try and figure out how you make the convincing argument that this device, which is far more expensive than a simple blood tube, how that is justified. And we all know there's other things that come into this, but the lab doesn't care that someone's saved on their gas bill or their public transport bill. They've still got a more expensive consumable that they've got to account for, and somewhere has to pay. And I think, this is one of the more complex parts of it. So, are there any solutions in this kind of area that people have been thinking about? Do we have silence for the first time? No solutions.
Waiting for a brilliant idea.
I don't have a brilliant idea. I think, unfortunately, the cost is going to be transferred over to the patient initially as they go for these newer technologies that are perhaps not as reimbursable, or, the shipping both ways has to be accounted for, the additional time in lab for handling all this. That cost is perhaps going to be transferred over to the patient initially, unfortunately. We just have to keep working towards getting general acceptance. And maybe at that point, then, having that reimbursement discussion may be a lot easier at this point. But to some degree, I mean, direct to consumer is a whole different thing, but it's kind of where a part of this field is. Not a lot of it, but it is a piece of this.
Yeah, I mean, as the capabilities, as they start moving towards clinical, the ability for somebody to be able to self-collect can take friction out of the process and improve compliance in terms of labs being performed. It also can be a game changer in rural markets where it's, you're in a healthcare desert. I'll use somebody else's term, not mine. But, it does have the ability to really impact public health or health. There's a value there, but, Brad, you're right. I mean, it's going to take a bit before, that, kind of the economics around this, the macroeconomics, can be addressed and appreciated.
So is there ways we can shortcut that to make that period faster? Can we develop, would examples of how to do cost benefit models be useful? Do we need to bring in some people that are good at doing cost benefit models?
I mean, the first thing I thought of when you said how to shortcut all of this, and I thought, well, COVID did a pretty good job of getting us headed in the right direction, unfortunately. But I wouldn't ask for another outbreak like that.
I was going to say that the vendors of these devices do a good job, I think, of pointing out that if it's a self-collection, then you've eliminated a whole cost center for phlebotomy and staffing, with benefits and so forth. And so for the collection part, I think that's a robust argument. I think the analytic part is the part that is borne, the increased labor costs are borne by the hospital or, the satellite lab or what have you. And that's the part that I think, the cost center there is different. That's what bears the cost, but then the benefits are accrued perhaps, in the system or perhaps on the patient side. And so unless you look at it holistically, it's tough. I think that from the UK perspective, there's a commitment to moving, it's a national commitment to move care more into the community. And so since that's a guiding principle and a consensus to work from, that's in concert with their, national goal, which is a 10-year plan. I think also you can approach it from maybe ADA and equity, healthcare equity model, where if these people are homebound, if they are cognitively, challenged and so forth, how do you provide care for them? And this is a way to do that. But those are special populations.
It's really interesting to think about offsetting the cost a bit with the elimination of the trained professional on the collection side. But I would also point out that you would want to make sure that your instructions and your success rate on a quality sample is really high, or else you could potentially lose that back on, repeat collections from the patient.
Yeah, good point. Yeah.
Yeah, I mean, the cost on it, they're different stakeholders, and it really is, it is a macroeconomic, challenge. You still need to have draw sites. You're not going to be eliminating draw sites, but tactically, it's really a kind of a tactical solution for people that are unable to get in or unwilling to get in to a draw site. In many cases, particularly for the rural markets, or maybe some homebound, and you can't run a mobile phlebotomist out.
So, is one of the solutions to maybe do some cost benefit analysis for certain situations? There was a PCSIG group that did some work on this, and we've just not had time to put the output of it up onto the website yet. But there's some really good thinking there of going through different use cases and trying to present the different aspects in one of those spider's web diagrams that just makes it quite clear that for this use case, you can see that overall it's beneficial, like the rural communities and communities with learning difficulties and other populations, maybe the benefit isn't as big. And working through some of those use cases and then having those out there, and would doing that help bring forward these technologies so communities become more familiar with them? And then, obviously once we manufacture more of these devices, the price starts to come down a little bit, and that starts to feed into other use cases. Is that how we start, or do we just dive in and do everything?
I think speaking about the different use cases and being, doing a better job of getting the word out as to how these devices, how this capability can be used will help, because it may trigger an aha moment for somebody who has a particular challenge within their own business environment. It's going to take time. This is still very, very early days in this I guess this segment of the laboratory market.
Yeah.
And then I wanted to jump in. Sorry, Neil, I'm kind of taking your role for a second. Gloria asked if any of us have been doing any publications. Brad has done some. We did something. We published a concordance study in obstetrics and gynecology on a series of fertility hormones with one of our partners, Modern Fertility. We did that back in 2019. And we do share our validation data with folks. I think that goes to one of the original or the points on the first slide with respect to providing some comfort with to the providers and to the patients. I think we need more studies done, a lot more studies done.
Yeah, I agree. And as I was saying earlier, it's not just doing the studies; it's then publishing the data.
Yes.
And companies not, I know, Brian, you're good at sharing. Brad, your group has been fantastic at sharing, and there's quite a few labs out there that have been doing that. But there's a lot of labs that haven't, and we need to find a way of encouraging them to share that data.
Well, I mean, to your point that you brought up earlier is now there's the Journal of Patient Centricity. Before, it has been a bit of a problem trying to find the right fit. There's a few clinical chemistry journals out there, but they have a high impact factor, and sometimes they're a little bit more difficult to get into. So having the Journal of Patient Centricity is actually a good step forward to have a place to put this and kind of consolidate it somewhat. And I know there's several more publications, not from my group, but from others that I've reviewed, that this is a rapidly growing area. There's data out there, and it'll be coming out in the next, probably by the end of the year.
Yeah, and I know a lot from European labs that are planning a lot of publications as well. So I think what we'll soon be moving into, I guess, is the the field of distilling down all those publications into reviews or
database of some sort.
Databases that's digestible. And, that's something that people asked PCSIG to take a look at, and we looked at it and it was just too overwhelming. And so what we ended up doing is writing a guidance and publishing it on our website, advising people how to do searches with AI tools. Just go and find your own data. And I believe, in the rapidly changing world we are with AI, so long as this data is placed in places that people can access using AI tools, it will be a faster way of you finding out than waiting for someone to publish a review article. So long as it's being put out there in reputable journals and it's acceptable, I think we need to find quicker and better ways of distilling that data than a review article.
And I will add a little bit to that. That data that you're describing from a regulatory perspective will be useful to fill the gaps that I was mentioning in the previous workshop last week regarding equivalency between matrices. And that's an important gap that is missing, that is not moving forward the actual authorization of the collection devices for use for testing. But that only takes you that far. That is the beginning for then the companies to do the studies for their own devices, to bring them into the market with claims that are based on solid evidence. So making studies here and there where details matters regarding what device was used, how sample was handled, and how what test was used, and how those pieces can be in isolation, be brought together to extrapolate on how we can generalize the data. That part needs to be addressed, because otherwise you will continue to have these little labs trying to do the best that they can in bringing forward solutions when you have manufacturers that I don't see them in the panel. I just realized we don't have manufacturers in the panel, and they have a big part on potentially moving faster this space.
Yeah, they definitely play a very significant role, and they know who they're selling to and potentially how the devices are being used. And as I said at the prior meeting, it's important for us within the community to self-police, to make sure that things are not being used inappropriately, which then can further degrade trust in the capabilities that a patient-collected sample can bring to the marketplace.
Right. So it's thinking about that data that you generate, that is the sample equivalency and the other aspects regarding the lab. Not every single piece will be figured out on its own, because they have to come together at some point so that you can have the puzzle complete. So definitely important on just generating data, but data with a purpose that have a clear target and goal. And of course, very biased as a regulator, to me the goal is authorized devices that labs can use. That's my goal.
Yeah. And Steve, I just seen Stephen, thank you for your comment. Good global and inclusive exchanges, thank you. We're getting to the end of our hour now. I think we've had some really great discussions. Just want to give everyone a moment. If there's something on the panel that was burning, comment that you never got the moment to get out. I think we've talked about a lot of good solutions here. We've got some good ideas for next steps with collaborative communities, bringing different groups together, making sure we include the voice of the patients, making sure we bring in the manufacturers and help them with the solutions. sharing information and coming together and sharing best practices is the kind of trends I'm hearing here. Is there anything else we're missing before we sum everything up?
I just have one thing to say, back to the patient-centric part of this. A lot of times in these studies, there's focus on, as is appropriate, the analytic, capillary versus venous. And, also for the FDA, you have to have the comparison with the predicate. But I thought that Brad did something really great in his recent paper where he actually gathered patient pain perceptions. And so since a lot of this patient-centric movement is related to more comfort and being more patient-centric, I think that that's a missing part from a lot of the literature. And Brad, do you want to say anything about that? Because I thought it was really excellent in that recent paper that you included that.
No, I appreciate it, and it's certainly a big part of the patient-centric, trying to understand, how all of these devices work. That they're not all the same. There's a lot of data out there to indicate that they produce similar results, but it's a piece of all of this, and I appreciate you commenting on it, Sharon.
Thank you, Sharon. Thank you, Brad. So just bringing everything together here. In the next few days, we'll be compiling all the comments that came online, all the discussions we were having with the panel here into a document that we'll publish within the PCSIG LinkedIn group for people to comment on. The panels from Europe and North America will be coming together to talk about next steps and making some proposals, which again, we'll bring back to the whole community for input on. And then we'll be asking you all in our community to take part in these solutions. They may be working groups, they may be publications, whatever, but we'll be asking people to step forward. We're a fully inclusive community and everyone's welcome to get involved. No one's excluded. And if there's voices we're missing, please invite them to come along. And saying that, for those of you that don't know about the patient-centric sampling interest group, please use the QR code on the left to visit our website. QR code on the right will take you to our LinkedIn group, which is open to everyone and is a good place to share information, ask questions, share your concerns, even just say, do people want to come and have a coffee when we're at a certain conference or in a certain town or whatever. It's there to help us all move forward, and we're approaching 700 members of that group now. So just like to thank our sponsors again for helping us to have this fantastic event today. I'd like to thank our panelists who, for such a great discussion today. We've covered a lot of ground today and last week, and unfortunately John couldn't be with us, but hopefully he'll be part of the ongoing conversation. And also, really importantly, you, the audience. We couldn't do this without you. And as I say, I want you all to be part of the solutions as well in the next steps moving forward. So no further ado, I'd like to close this event. The recording will be available through our website and eventually through our YouTube channel. Thank you all for your attention and for attending today.